What we've got here is what I'm calling a bit of informed speculation, grounded by many individual pieces of good science that's mostly been done through Norm and his colleagues at Queen's University. But Norm and I have put this together into kind of a complicated spreadsheet of what we think, how we think we might have to start interpreting algal blooms in the future. This is a talk that I gave at the Canadian Ecotoxicity Workshop in Halifax last October. So as we probably all know, algal blooms are on the rise in our Great Lakes, our Precambrian Shield Lakes. This is data from the Ministry of the Environment, Dorset people, going back to 1994, and shows really that over the last 20 plus years, we are seeing an increase in the number of confirmed algal blooms, and especially those which are the lightest color, which are dominated by cyanobacteria or blue-green algae. These are the algae that are the most unpleasant looking on the surface of the lake and which can occasionally generate toxins. Not pleasant by any stretch of the imagination. Most of the algae that is beneficial belong to other genera. The weird thing that we're seeing, though, is that these algal blooms we've often thought are a function of human presence and human development on lakes, but we're seeing them on remote lakes with no human habitation at all. And this is a bit of a mystery and kind of troubling. We've been thinking about algal blooms in one particular manner for 50 years or longer, and we're now faced with algal blooms where we don't think they should be, and the question is why? Norm, feel free to speak up at any time. We thought they understood the cause of algal blooms was too much human phosphorus, anthropogenic phosphorus. This is a classic picture from limnology. David Schindler and his colleagues in 1977. People were not convinced that phosphorus was causing the algae problems we were seeing in the Great Lakes at the time in the 60s and the 70s. Dr. Schindler put a curtain across the Narrows on Lake 226 and put carbon, nitrogen, and phosphorus, the three prime nutrients, on one side, and just carbon and nitrogen on the other. And lo and behold, the side with the phosphorus turned soupy green. A definitive experiment was this photograph was on the cover of the Science Journal, the most prestigious journal out there at the time. This was a definitive study. So we've been focused on phosphorus as the causative ingredient for algal blooms for a long time. And it's certainly true. Too much phosphorus is a concern. But what I'd like to point out is it's usually a concern at much higher levels than what we see in Muskoka. The top slide here is Peter Dillon's PhD thesis and Dillon and Rigler showing a great straight line relationship between chlorophyll, which is the green pigment in algae, and phosphorus across the bottom. As you add phosphorus, chlorophyll responds. But you'll notice that the graph goes from 10 parts per billion to 100 parts per billion to, well, let's say 300 parts per billion. Whereas the lakes that we're worried about are pretty much less than 10. So yes, there's a relationship, but it doesn't kick in until higher levels. Here's some work on Lake Okeechobee in Florida, which shows that mean chlorophyll, again, increasing with phosphorus, but again, at concentrations above 10 parts per billion, closer to 20, where and then you see the frequency of a bloom, which here is more than 40 parts per billion of chlorophyll. You really start to see blooms happening frequently when you get up to the 20 plus 40 part per billion. So blooms are happening, and we've always known that with phosphorus, but at much higher levels. And this is another graph from Minnesota showing the same thing, that your frequency of blooms from low frequency here, low intensity blooms, the darker it is, the more algae there is, increases with phosphorus and increase in the frequency with which they occur. But really, it's over 20 is the problem. And so this has caused us quite a bit of concern in Muskoka. But nevertheless, for 50 years, the paradigm on algal bloom causation has been it's all about the phosphorus. In Muskoka, that's been reflected in our official plans in the past by looking at development and how it influences phosphorus loads through sewage plants, septic systems, and urban runoff, wastewater, increasing phosphorus, or by land use, clearing the land, cutting the forests, adding fertilizer. getting more phosphorus into the lake as you cut the trees down and put farm fields in instead. You're going to get less phosphorus uptake and more into the lakes. And this is how we've been managing water quality for 50 plus years. But the one thing that we're seeing, in spite of the fact that we're seeing more blooms, we've done a pretty good job on managing phosphorus. Our classic example is Gravenhurst Bay, which was up in algal bloom country, used to get algal blooms up in the 20 and 40 parts per billion of phosphorus back in the 70s, until they started treating the phosphorus in the sewage outfall that goes into Gravenhurst Bay. And then around about mid-90s, moving the outfall into the main basin of Lake Muskoka to continue the downward trend. A nice aspect of recovery there. We also have to recognize that Muskoka's lakes tend to be low nutrients. Here's the district's data on 196 lakes going back to 2014, where most of our lakes are in the 8 to 10 parts per billion range. Lake Joseph, lakes like that are down at 4. Brandy Lake, Halfway Lake, where I live, are up in the 18 to 20 range. But we have very low phosphorus lakes. The district has been recording blooms in Muskoka lakes and initiating causation studies. And here's an example of lakes where algal blooms have been observed in the last 25 years, and more specifically in the last 5 years, where we're seeing blooms as low as 5.6 micrograms per liter or 5.9, all the way up to Brandy Lake with 17, or 3 Mile Lake in the main basin at 17. But these are all below a 20 microgram per liter Ontario water quality objective for protection against algal blooms. The province of Ontario is at 20 parts per billion of phosphorus. Is a cap. You don't want to go beyond that, or you'll risk algal blooms. But so our lakes are getting blooms, but they don't have as much phosphorus as we think they should to generate a bloom. So is there anything else? Are the new lake stressors? Is climate having an issue, doing something to cause these new algal blooms that we haven't seen in the past in these lakes? Well, there's 3 factors that can increase your risk of algal blooms. They're influenced by the species preferences and tolerances of lake chemistry and physics, the habitat does the lake a good habitat for bloom-forming species? Are they linked to faster algal growth rates because there's more phosphorus in the lakes? And the third factor is the death of algae, that is, they're grazing by zooplankton, which tends to clear the water. Norm, I'm sure, has told us in the past about the incredible filtration that cladocera and zooplankton undertake in our lakes, keeping them clean. Well, if something was to happen to harm the zooplankton, you would have lower algal grazing, and that might cause a problem. So what we've done here is assembled information relating to each of these, so we call them hypotheses. So there's 2 pathways we've considered. One is a pathway of nutrient enrichment, which is the classic paradigm of phosphorus enrichment. The other pathway is toxicological responses that might impair zooplankton grazing. We've looked at 5 potential stressors, all of which have been documented in our lakes: acid rain, climate change, calcium loss, climate change again on both sides of the equation, or chloride toxicity, and wondering if looking at these in kind of a cumulative effects scenario might help explain why the algal blooms are happening. So again, the nutrient enrichment, this is the paradigm we've been working on for the last 50 years, which simply says if you add phosphorus, you're going to increase the load, faster algal growth, increasing risk. Or if you clear the land, add fertilizer to it, you'll add a load of phosphorus for faster algal growth, increasing algal blooms. What happens if you add acid rain as a stressor to this, though? If in fact acid rain, well, not if, it does impair tree growth directly by the acidification and changes in chemistry of the soil. If you impair your tree health, you have a leakier watershed. The forest is not as able to suck up phosphorus as it was before. There's less phosphorus sequestration, and so the phosphorus that you are generating from land use is more likely to end up in the lake and increase your risk of an algal bloom. So acid rain kind of works on the nutrient enrichment pathway. So what happens if we add a warming climate to this? So if we get climate change, the temperature of the water goes up, the temperature of the soils go up. Microbes can increase the rate at which they mineralize phosphorus from non-available into available forms and result in an increased load of available phosphorus just by the fact that it's getting warmer. Temperature increases too much. The trees here, which are adapted to climate in years past, would have their health reduced by the increased warmth, would reduce their ability to take up phosphorus, and again, increase the load of phosphorus. The other thing we're seeing with climate change is increased storm severity. When the storms happen, they tend to be more severe, more violent. That could increase the erosion, which, again, if you've got reduced uptake, sequestration, and reduced trees to help absorb the energy of the rainstorm, you would end up, again, with more phosphorus coming into the lake. So there's one aspect of a warming climate that could be several mechanisms by which it could be moving more phosphorus into the lakes. Okay, sorry, that's my problem with PowerPoint. The final climate change scenario here involves increased temperatures, which will increase the stability of our water columns. We have stratified lakes here, warm water on the top in the summer, cold water, denser water underneath in the summer, and these two layers don't mix. If the lakes warm up earlier and the ice comes off earlier, your stratification is going to set up earlier and your lakes will be more stable. What you get over the summer is algal growth falling out of the top of the water column into the bottom and being consumed by bacteria, which take the oxygen out of the water. And that's a problem for cold water, high oxygen requiring fish like lake trout. But if you take out all of the oxygen from the bottom, you get an internal loading of phosphorus. So you get more phosphorus comes into the bottom waters and increases the available load of phosphorus. So we think this might be quite an important pathway for climate change to change the phosphorus dynamics in a lake. You end up with more load in the lake. The other thing you get, though, is some blue-green formers, cyanobacteria, can migrate vertically. If the water column is stable and still, they can sink down to the bottom, suck up all this extra phosphorus from the internal load, and then pop back up to the surface where there's sunlight and have a bloom. So these are
Neil,
other things that we have to start. Yes, Norm.
Can I just add one comment there? We're not suggesting this will happen in every single lake, but this is the actual mechanism that was proposed for Dixon Lake and Algonquin Park. Longer summer stratification, more anoxia, and then a late summer, early fall bloom. But across Muskoka, we're not, I don't think, generally seeing increased anoxia, at least not in the Dorset long-term study lakes. But absolutely, this so everything is going to be a bit lake specific in terms of which of these mechanisms might be most likely. But this is the one that seems to have been suggested for Dixon Lake and Algonquin Park.
Thanks, Norm, and that's a good clarification. And I'd add on top of that that the district's data and the Ministry of Environment data both show that phosphorus levels are not increasing in our lakes in Muskoka. They're stable, or if anything, decreasing. So some of these, like storm severity, could be short-term events that don't show up in a spring overturn sample. Internal loading of phosphorus would not show up in a spring overturn sample to any great extent. But these are all ways that at certain times we could see that there could be a link to algal blooms. The other thing is blue-greens like stable water, as I said, stable warm water. Ministry of Environment data from Dorset shows that at the bottom, wind speeds of going up to 1.4 to 2.8 meters per second, average wind speeds have been dropping. Higher winds do not occur as often as they did in the past in Muskoka lakes, whereas you're getting more days with a low wind speed, less than 1.4 meters per second. So the lakes are getting calmer and more stable. So this provides better habitat for cyanobacteria. Algae like diatoms are heavier and will sink out of the surface waters unless the waters are turbulent, which will keep them mixed up and up in the sunlit layers. but as the water is calmer, they'll sink away from the light, whereas blue-greens can regulate their buoyancy and can stay up where the sunshine is.
I'll also add, Neil, that photograph of the Bloom and Dixon Lake happened in the year with the lowest wind speed on this graph, 2014.
Excellent. I mean, good connection, Norm. Yes. So we've got the same climate signals we had before of storm severity, warmer water, lower wind speed, translating into better blue-green habitat, which provides for more algal growth. So algal blooms are on the rise, and they can occur in remote lakes. So this is the nutrient data suggests this, and the nutrient mechanisms. So we know that nutrient enrichment and favorable habitat could both increase the likelihood of algal blooms. Let's talk about toxicological responses. Because as Norm will tell you, algal growth is controlled by zooplankton grazing, and if you have a lower algal death rate, you can have increasing risk of algal blooms if in fact something is happening to your filter feeders and your cladocera. We all know, I'm sure, Norm, you've presented this in the past to the Watershed Council, of the importance of calcium to our freshwater system. Our good filter feeders like Daphnia are 5% calcium. Lots of calcium in loon eggs and turtles and crayfish and fish and even us. Calcium is an essential nutrient. But we know that calcium is declining in our lakes back over the years of acid rain from 1980, which I'm sure acid rain was actually started 20 years before that, so we probably had even greater declines if we go there. But we lost 40% of the calcium in our lakes in Muskoka on average. These are 7 of the lakes in Dorset in the last 40 years. So we've lost the calcium. There's less calcium in the water. And of course, if we run some bioassays that Norm and his colleagues did out at Dorset, we'll see that if you get down to 0.1 milligram per liter, Daphnia simply stop reproducing. They do fine if you have, again, I'm having trouble here, 1 milligram per liter or higher of calcium in the water. The reproduction is not impaired. They're reproducing just fine. But as you decrease the calcium, you decrease the reproduction, the ability of Daphnia to reproduce. Oh, go back here. So you can have acid rain reducing your tree health, increasing your loading of phosphorus, but you can also have a calcium deficit in forest soils, declining calcium in the lakes, reduced growth, reproduction, reduced grazing, lower algal death, increased risk of algal blooms. So that's the basic mechanism of calcium loss, but now we want to look at how these things might interact together. We know that high temperature reduced the growth of Daphnia. This is again some of Norm's work, and we see that the growth rate of Daphnia really starts to decline at about 29 degrees. Most of our lakes in the past have been in the 23 to 26 degree range up in the surface waters in the summer. But if you get up to 29 or higher, the growth rate declines. And then the calcium effect on reproduction is also increased as water warms. So here's 1 milligram per liter of calcium at 20 degrees, at 24, and at 28 degrees, showing reduced reproduction as the water warms and the calcium decreases. Well, is 29 degrees reasonable? We were out swimming in my lake, which is a small, still brown lake, last August, and we're remarking how warm it was. So I went into the lab and got a meter, and we were near 32 degrees up at the surface of this small lake. Of course, it declines with depth, but the upper 30 centimeters of the water were in fact at 29 degrees or higher, not very good for Daphnia. Now, of course, if you measure oxygen, you'll find that once we got below 1 meter in depth, we had less than 4 parts per million of oxygen, declining to no oxygen in the bottom meter of the lake water. This is just the demand existed by the sediments. This lake is too shallow to stratify, but there's quite an oxygen demand. It's a shallow lake. So we have hot water at the surface and low oxygen water at the bottom, which really squeezes the Daphnia and the filter feeders into a very small volume of water of about 0.5 meter depth. Below that, there's not enough oxygen. Above that, it's too hot. So as the climate warms, we're seeing more potential that algal growth and reproduction will be impaired and their habitat will be reduced, increasing decreasing the grazing and increasing the risk of algal blooms. So we've looked at climate change again. Now we want to look at chloride. Again, this is not news to anybody in the Watershed Council. We add enormous amounts of salt to our landscape every year so that people can drive too fast in the wintertime. And much of that ends up into our lakes because it's a conservative element. Here is Lake Muskoka at the bottom, the main bays in Lake Muskoka, all showing going back into the 80s from MOE and Dorset data, increasing chloride from 2 or 3 milligrams per liter up to 6, so that's a doubling. In Gravenhurst Bay, which is much more urbanized and a smaller water body, we've gone from 6 or 7 up to 16. And I'd point out that Gravenhurst Bay probably started down at 2 or 3 like Lake Muskoka did at one point. We just have been measuring since then. There's 16,500 tons of chloride in Lake Muskoka, with a surface area of 12,000 hectares and a drainage area of 460,000. Now, Norm, you have done a calculation on how many bags of road salt that equates to.
Yes. How many bags of road salt? Well, it's about a ton of salt for every permanent resident in Muskoka is actually added to our road. So if you go into the grocery store and see a pallet of salt in the winter, they tend to be 25 bags of 10 kilograms each. So that's the equivalent of 4 full pallets of salt is roughly the amount of salt that's added for every permanent resident of Muskoka to the watershed, so that, as Neil says, we can drive too fast in the winter.
So, and we're seeing that increase in the lakes. And this is showing that 100% of our lakes here have less than 110 milligrams per liter of chloride. 50% of the lakes have less than 10, which is probably where you want to be, and we'll get into that in a minute. But most of our lakes probably started off again in the 0 to 5 milligram per liter chloride range and have been increasing over time, and the number of lakes that have high chloride. So we're seeing lots of our lakes being enriched by chloride from the runoff from our roads and our parking lots and our driveways.
Yeah, let's just acknowledge Rebecca for those data. Thank you, Rebecca.
Great. The federal government in 2001 declared road salt as a hazardous substance, and over the years developed the Canadian Water Quality Guideline, CWQC, for chloride of 120 milligrams per liter, suggesting that concentration was good for indefinite survival of aquatic life in Canadian waters. So just keep that 120 in mind. It looks good. All of our lakes in Muskoka are less than that if you look at 120. And in 822 Ontario lakes, again, much as we saw in Muskoka, 75% of the lakes are less than 5, 23% 5 to 40, and the remainder are up in the high range as well. Maybe 5 or 10% are up in the 100 part per million range. But let's look at what happens to road salt as our climate warms up. This is a bit of simple modeling that I did, where I took measured data from December to February of 2017-18 and plotted that in the light green here, and showed that 6 times over those 3 months we went through 0 degrees. So this is what would happen if you're going from warm to cold, is where ice would form, and it would melt again, form again, melt again, form again. And these are the periods when we'd expect to be adding lots of salt to the road because you're going through a freeze-thaw cycle. The federal government, the Department of Natural Resources, the modeling shows we can expect an increase of 3.5 degrees in our average winter temperature between 2031 and 2050. What that would do is just displace this temperature cycle up, and you would have 13 of these episodes where you went through a freeze-thaw cycle in the future versus 6 of what we've seen in the past. Again, this suggests that as the climate warms, it's not going to make less ice. It's because we're still under zero a lot of the times. We're going to have more episodes where ice would form and salt would have to be applied. So here's the Canadian Water Quality Guideline of 120 milligrams per liter of chloride. It's designed to protect 95% of the aquatic life that was tested. On the right here is quite happy at 120 milligrams per liter or higher, but at 5% are suffering at under 120, but we choose the fifth percentile here as our water quality guideline to protect 95% of our species. So this is where the magic number of 120 comes from. These are all lab studies. That's the water quality guideline for chronic long-term exposure, continuous exposure, whereas 640 parts per million will kill something within several days. So let's now compare this to some of the more recent data that Norm and his colleagues have been doing. Did you want to talk to this slide, Norm? The next couple slides?
Sure, if you'd like, Neil. So as Neil said, that previous slide was done entirely in the lab with growth conditions optimized for every one of those species. And then chloride was added because the toxicologists are really only interested in the effects of chloride. The reality, so it's kind of like couch potato toxicity studies for, happy organisms. Make them as happy as possible and then pull away the couch and see how often you have to do that with chloride to kill them. But lakes aren't like that. So with Shelley Arnott and Martha Salazar, my former postdoc, and others, Shelley has led studies, continuing work I did with a master's student, Aaron Brown, a few years ago to ask, well, what if the work was done at calcium levels that are typical of Muskoka lakes, not calcium levels that make Daphnia happy? And when you do that, you see that there's very dramatic reduction in productivity, that's the number of neonates, at chloride concentrations well below 120 milligrams per liter. This is still lab work, but Daphnia mendoti, the graph on the right, that's the most common Daphnia species we have in our lakes. And at the lowest lake calcium level, that was, and calcium levels typical of Muskoka lakes, there's dramatic reductions in fecundity at the lowest chloride level. In fact, at 120, there was almost complete elimination of production of babies by this Daphnia species. So it looks like if you just work in the lab at conditions that are typical of Muskoka lakes, the 120 milligram per liter level is not protective of our waters. And it's not just low calcium that might make road salt more sensitive. It's actually the very oligotrophic nature of our lakes. All previous lab studies are done at optimal food levels and optimal calcium levels. So a student, actually my last graduate student before I retired, Aaron Brown, asked the question, what if we worked at food levels that are typical of Muskoka lakes? Would we get a different idea of sensitivity to chloride, either presented as sodium or calcium chloride? And so these results show a linear response between the amount of food that the animals have and chloride sensitivity. And So Neil, does the next graph show where those lakes are? Did you add those? I see plastic, red chalk, blue chalk. Oh, I see. Yes, there they are.
Yeah.
So you'll see that most of Dorset's long-term study lakes have all but one, all but chub, have food levels that lead to sensitive chloride exposures well below the 120 milligrams per liter. In fact, for plastic and red chalk lakes, it suggests that roughly 40 milligrams per liter of chloride would kill these animals. So we're not looking at kind of we're looking at quite a serious consequence, which in this case is killing half the animals in a 2-week experiment. You want to take over again, Neil?
Okay, so the upshot of what Norm has said is again, we saw Rebecca's data on the distribution of chloride and the increases in chloride in our Muskoka lakes. The Canadian Water Quality Guideline, as I said earlier, would protect our lakes if we based it on that. But if we started looking at some of the more sensitive responses that Norm and his colleagues looked at, 40 milligrams per liter would still, which lots of our lakes are, is maybe safe for some, but for the total safety factor for the most food-starved, low-calcium lakes, that water quality guideline should probably be closer to 10 milligrams per liter to protect filter feeders, these zooplankton species in our low-calcium lakes. So this really changes our vision of things. We can't pretend that 120, because we're under that, we're safe, when in fact somewhere between 10 and 40 we're seeing the potential for significant damage. And it's not just the potential for damage. This has been realized. This is a complex slide, but Jevons Lake, if you're coming north on Highway 11, you go through to the great big curve on Highway 11, and that passes right over the inflow to Jevons Lake, which is downstream, just a couple hundred meters from the highway, and is at 105 milligrams per liter of chloride. It's receiving urban runoff from the town of Gravenhurst. All the parking lots of the McDonald's and the plazas over here drain down through this little wetland. They pick up more going from the Highway 11, and Jevons Lake is increasing. Friends of Muskoka Watershed have a continuous monitor installed right downstream of the highway, and we're monitoring conductivity chloride levels going into Jevons Lake. But if we compare that to Heaney Lake, which has got 1 milligram per liter of chloride, we see through ordination and statistical space, this representation really means that you have a dramatic change in the aquatic community of Jevons Lake that's preserved in the lake sediments, starting at about 1950. Right about the time we started really socking the salt to the roads, the zooplankton assemblages changed, mostly through the loss of this one species, Bosmina, compared to Heaney Lake, where that has not happened. So the potential that we see in the toxicity experiments is realized in the field. Go ahead, Norm.
Yeah, I'm just going to add, I think this is the first demonstration in Canada of the impacts of road salt on an entire lake assemblage of species, this work in Jevons Lake.
So we see all kinds of things working together to increase the risk of algal blooms. We have nutrient enrichment from acid rain, from climate change. We have habitat changes related to climate change. And we have toxicological responses of lower calcium, warmer temperatures, and chloride toxicity all interacting to provide a cumulative effect of potential of all these 5 different pathways to increase the density of algae, but more importantly, improving, damaging the zooplankton that feed on algae. We have more salt in the water, it's warmer, increasing damage because the lakes have lost their calcium from acid rain. Increasing nutrient supply, more erosion from the flooding systems, potential for internal loading of phosphorus, and warmer water, improving the habitat for blue-green algae. Warmer water, lower wind speeds, later fall turnover, lower bottom water oxygen, late summer and fall. So it means Every one of those arrows there is probably somebody's master's student, master's project that should be undertaken to look at this more thoroughly. As I've said, these are all pathways and mechanisms that have been demonstrated and backed up by field evidence, but linking the whole thing together becomes quite stable or quite a challenge. But it means that we shouldn't just be focusing on increased loading of phosphorus from development as the only problem that we can manage in our lakes. We're going to have to increase the depth at which we look at the causes of these algal blooms. And we can no longer just simply say that it's the number of cottages on the lake, for example.
Can I add
well, thank you very much.
Neil, can I just add 2 things to that slide? If you can go back to that slide. Some of these things are very counterintuitive. So for example, I never would have thought that average wind speeds would be falling while at the same time storms could become more severe, but that's in fact what's happening. And then despite this complexity, it doesn't include everything. So for example, the spiny water flea is not on this graph, but it's spreading around Muskoka and is decreasing zooplankton populations. And so it probably should be added on the right as something else that will decrease zooplankton populations. And these things, as I said before, don't happen in every lake. But I think more commonly, we're having a couple of them co-occur in every lake. And that's where things get a little scary for me. So for example, in one of the ministry's long-term study lakes, Harp Lake, calcium levels won't have fallen enough yet to stress zooplankton, but the spiny water flea are there. And so it's this idea that you need to kind of look at not at every lake, but realize that there might be multiple things or more than one thing happening in particular lakes that will actually, Neil and I think, might increase the risk of algal blooms. But this is just a big, as Neil said, kind of hypothesis map at this point. I know Liz Favat is considering some of these things in her postdoctoral project that she's doing I guess in part with FOCA. But there's years of work to be done here. And I think Neil and I just hope to set the stage for for what we think of as logical possible linkages to increased algal blooms. We can't ignore it because the reality is, as Neil said, our model of the last 50 years, which is that it's just all about spraying average phosphorus concentrations, no longer seems to be the case.
Thanks, Norm. Yeah, and I'm just pleased that Norm has asked me to work with him on this. So much of this stems from the research that he did. And we are drafting a manuscript for publication in the literature that hopefully we got one draft done, and we'll be working on that over the course of this year. So thanks, Peter, and thanks to the Watershed Council.